Well, I'm really happy to be here, and this is my 33rd talk on pawpaw in the last 7 months. I'm just about shot. But I'm really happy to talk about it anytime, any place. And it's been my life's work. And I'd like to be able to spend the next hour or so to tell you about it. The next slide is a disclaimer slide, which is necessitated by the bureaucratic society that we've created for ourselves. I'm not supposed to use the word cancer. And yet 1 guy in 2 and 1 woman in 3 out there is going to get cancer. It's Big Pharma's business to talk about cancer treatments or cancer cures. Okay. I was watching the public television show, the Red Green Show. Did you ever watch that? He's got a red suspender and a green suspender. The other night he was signing off and he said, well, goodbye out there, all you folks. And I remember keep thinking out there, because if we don't think, government happens. And I had, in February, I was on a radio show out of Salt Lake City. Dr. Hugo Rodier has a medical talk show and had me on and said, well, what's your opinion of the Food and Drug Administration? And a lot of times I forget that I'm on the air. And I said, well, I said, Hugo, we can all rest assured that the Food and Drug Administration will be there protecting our health as they watch us die. And that's exactly what we have today. We're over-regulated, okay? So I have to put this up. 3 lawyers and 4 lawyers' assistants have been over this talk, okay? The next slide. I want to give you some facts and figures. These are readily available from the American Cancer Society, and I don't want to go over these all, but I just want to point out the third one down there is about 600,000 people die of cancer per year in the United States. 600,000 people. That's 1 person a minute that dies of cancer in the United States. 1 person a minute. To put that number in perspective, in the Civil War, in 4 years, from 1861 to 65, we lost 600,000 men on both sides. And that was in 4 years of the Civil War. But in 1 year, we lose that many people to cancer. And in the Vietnamese War, we lost 60,000. In all the years, from the 60s into the 70s in the Vietnamese War, 60,000. So each year, 10 times the number of people who died in the Vietnam War die of cancer. And we lost about 3,000 people on September 11th due to the terrorist activities. 3,000 people. 600,000 people died that year of cancer. And because of those 3,000 people, we invaded Afghanistan and Iraq. And we're ready to spend $87 billion to try and restore Iraq. Now, President Nixon declared war against cancer back in 1971 or 72. Whatever happened to that war? Did we win it? I don't think so, because I just read in USA Today this summer that cancer cases are up 19% since 1990. So we didn't win. What's happened to that war? We spend about $2 billion a year, as I understand, through research at the National Cancer Institute, and that's good. But unfortunately, we've left it up to big pharma to develop treatments for cancer. And a lot of the money that's gone into cancer research at the National Institutes of Health has gone into the study of the biology of cancer. So we've paid for the Human Genome Project. So we understand all the genes in the body and all that stuff. And this has helped. We understand the biology of cancer a lot better now. But what about those folks, those one guy in 2, one woman in 3, that got cancer or are going to get it? We haven't really served them well. Okay? The next slide. The physician has 3 treatments, 3 modalities to try and attack cancer if you've got cancer. Radiation and surgery have been around for a while. With surgery, of course, it's logical. If you can cut out the tumor and you have entire margins around the tumor where you don't cut through any tumors, any tumor cells, and you get rid of it, and it's the primary tumor site and it hasn't metastasized or spread, then you can get rid of it. But if you cut through the tumor, there's a possibility that cancer cells will break away from that site and can get into your lymph and can cause cancer a little bit later. Even if you do a biopsy and go into that tumor with a needle and disrupt some cells and pull them out, the lymph system can pick those cells up, and your lymph nodes distant from that site will develop the tumor later. And so the biopsy itself can spread the cancer. So surgery certainly isn't the answer. Radiation uses ionizing rays to go through cells and scramble the DNA so that the cells can't reproduce themselves anymore. Okay? So radiation can work, but you can have a hard time focusing radiation exactly on just the tumor cells. Okay? They hit other cells. They certainly have to go through good cells to get to the bad cells. Okay? And so that really isn't perfect either. And of course we know that 3 things cause cancer. There's physical things like radiation that causes cancer, okay? And then there are biological things like certain viruses, Epstein-Barr viruses associated with Burkitt's lymphoma, and then there are chemical things, the carcinogens. We know definitely that benzoate pyrene in tobacco smoke causes cancer. It binds onto the DNA and changes the DNA, changes the bases, and you end up causing cancer with the components of tobacco smoke. That's definitely there. But other chemicals, you know, in my research work for my PhD, I worked with a compound called benzidine. And now that's been shown to be cancerous. And I'm seeing some funny reactions now on my urine samples for bladder cancer, and that causes bladder cancer. So, you know, chemicals cause cancer. So all those things cause cancer. So radiation certainly isn't an answer. And chemotherapy then is the last thing. This came to us actually after World War II as a result of chemical warfare with nitrogen mustard studies. Remember in World War I, they used nitrogen and sulfur mustards as war gases. And they found that, that nitrogen mustard will cross-link the DNA strands and keep them from unwinding so that you can't replicate the cells. And so this cross-linking, or alkylation it's called, with these alkylating agents, that led us then to a whole series of 12,000 compounds that were all synthesized based on the structure of nitrogen mustard. Okay? And there's very little innovation. They're all based on that one structure. And the whole thing was based on a toxic action in the first place. So chemotherapy started right from the beginning being toxic. And it still is. And the old adage, the oath of Hippocrates, is, "If physician first do no harm," I don't think they take that oath anymore. Because if you do chemotherapy, you're going to do harm. Right? And this is the way the chemicals have evolved that we use for chemotherapy, because they evolved from harmful chemicals, cytotoxic chemicals that kill cells. So the antimetabolites like folic acid, methotrexate's an example there. Folic acid, you know, is a vitamin. So methotrexate's an antimetabolite of folic acid. So your cells take it in, think that it's folic acid, but it doesn't work. And it messes up the cells. Tubulin inhibitors. These are like from the vinca plant, the periwinkle plant, vincristine and vinblastine. They prevent the cells from separating their chromosomes at metaphase, so that the cells all sit there with their chromosomes duplicated, but they can't pull them apart to make the daughter cells. And so topoisomerase is a repair enzyme, and you can go on with adriamycin and other types of anti-cancer agents that almost all cause cancer. 10 to 15% of patients in long-term on chemo develop tumors from their chemo. So anti-cancer agents, the typical ones that are used and are sold by big pharma, cause cancer. No doubt about it. Okay, and then there are the side effects of the chemo. They're not selective enough. Selectivity means that we attack usually the rapidly dividing cells of the body. And normally we have rapidly dividing cells in our hair. The roots of the hair are rapidly dividing. So you take a chemotherapy agent and your hair falls out. Spermatogenesis, you know. These anti-cancer drugs would be really good male anti-fertility agents. They decrease sperm counts. Of course, guys wouldn't feel like having sex anyhow, so it would work well. The bone marrow is continually replacing itself. And there are 3 types of cells in the bone marrow that are made there. The blood platelets, they're little tiny ones. They have to do with clotting the blood. So if you're on chemo, you have a pink toothbrush because your blood won't clot and your gums are bleeding. And sometimes you have to go in and have platelets transfused from other people's blood. And then you develop allergic reactions to it because your immune system rejects those other people's platelets, just like you get the wrong type of blood cells, right? Well, okay, then we'll give you antihistamines so that you don't break out in hives. We got a whole series of drugs today that have been developed to attack the side effects of chemo. I bet tonight on Dan Rather there was somebody that was tired from chemo and they were pushing erythropoietin, which is a compound that builds up your blood cells, your red cells. So red cell depletion goes along with bone marrow depression. And so there's a big market now for these drugs to attack the side effects of chemo, right? Is that right? And then the white cells are depleted. And the white cells are the body's policemen. These are the cells that we have. These make up our immune system, and they help to reject foreign things like infections, viruses and stuff that come into our body, or cancers, as we develop cancers. So we go in here with chemo and poison the very mechanism that we have to reject the tumor in the first place. Does that make any sense? No. And then you get infections. Well, then we got antibiotics, right? So people on chemo get antibiotics. So we need an improvement, and we need a different type of thinking in big pharma on what type of drugs to develop, what type of mechanisms, what type of attack can we take to try and get at cancer, okay? And we just don't see that because the people there all base their thinking on what's already there. And the whole idea of chemotherapy has been based on toxic things. So we're developing more toxic things. The next slide. Okay, I was at Purdue as a professor for 28 years. I served 34 years in total as a professor, and I retired in 1999. And at Purdue, I did several things that I'm very pleased with. I developed a couple biological assays that you used to detect anti-cancer activity. And you don't have to spend a lot of money to use these assays. One of them is using sea monkeys. These are called brine shrimp. And you can grow these and hatch them from the eggs and grow them in salt water, and you can kill them and nobody cares. They're not protected by Senator Dole's law in 1984, which gave animals rights and welfare. That law set cancer research back at least 20 years. We were using about 500,000 mice a year in an anti-leukemic scheme. And suddenly, the National Cancer Institute, in order to get a better appropriation, said we won't use animals to do cancer research. Boy, was that dumb. But that's what they did. So I developed an assay using brine shrimp. Brine shrimp are very sensitive to things that in their water that they're swimming in, and all anti-cancer agents kill brine shrimp. So I said, hey, I'll just kill brine shrimp and look for things that might be anti-cancer in plants. Second thing I did is to get a little more specific. I grew tumors on potatoes. Plants get tumors. And I called this the potato disc assay. And I could inhibit the growth of tumors on potatoes with anti-cancer agents. And so I could use that then as a secondary screen to follow up my brine shrimp positives. And then I would go to Purdue's cell culture lab where they had 6 tumor cell lines from human tumors growing, and I could have my actives then screened in that assay, and that cost $120. And boy, that will burn up your research funds real fast. So I could do my biological assays with real cheap biological assays without having to go to cell culture and burn up my money. I could spend my money on the chemistry of my plants instead of on the biologist. Okay, so that was my approach. I had actually support from the National Cancer Institute for 24 years. And the work that I'm going to talk about tonight was supported by $5 million of your money. $5 million bucks. Now, that's not really quite true because the university has what they call indirect costs, and the administrators take a third of your money away from you so that you don't have that to spend. And at Purdue, our president needed a jet airplane. Now, during this time then, I was able to secure 4,000 species of plants. Now, this is a lot of plants. In all of the United States, there are only 6,000 species of plants growing. So I had 4,000 species of plants that I personally had my hands on, and I got through the screening of 3,500. So I had 500 left that I couldn't get through, and I gave those to the National Cancer Institute, so they're screening them now, so that's being done, okay? And out of those 3,500 species of plants, I had plants from Central America, South America, Mexico, North America, Africa, Asia, Australia, you name it. I went through all those plants one by one, and guess what the best one was? Paw-From, growing 2 miles from my office. Okay? So there it is, growing in your backyard, the best thing that I ran into for cancer. Now there are some relatives of Paw-From that are tropical that are equally potent, but by gosh, this one was right there, and we have plenty of it to work on. And incidentally, if you try to work on plants from somebody else's country today, you're accused of being a biopirate because you're stealing their resource, okay? And so this has really put the crimp on doing research on plants from the tropics or from other parts of the world because all the politicians want to be involved, and you have to have contracts signed on who's going to get the benefit if anything is found. And by the time you get all this stuff done, you know, you spent so much time just doing that you don't have any time to do any research. You know, it's so stupid how greedy people can get. In all these plants I isolated about 350 new compounds that were significantly cytotoxic in our Purdue cell culture lines. And these were new compounds. So that's a lot of work. So this is what I'm good at. I can do grind and find research. I can grind up a plant and find what's in there and tell you if it's biologically active, okay? And the Annonaceous acetogenins then are the most important leads. And these are the type of compounds I found in Paw-From because the plant family there is called the Annonaceae. And so these are called the Annonaceous acetogenins, aceto referring to acetate, that's like vinegar, And so these are compounds that are put together with 2 carbon units of acetate into long-chain fatty acids. The next slide. So this shows us a picture of pawpaws, a little cluster of them called the Indiana banana. And when I was about 4 years old, my dad gave me some of these and said, Jerry, you can eat these. These are Indiana bananas. And I was hungry for bananas because in World War II, you couldn't get bananas. And I ate a whole bunch of them and I threw up. And I threw up. And I never forgot this, and I had it in for this plant for a long, long time. You know, I knew there was something in there. Because if you throw up from eating something, you know there's something biologically active there. Okay. The fruits of them taste sort of like banana custard, and a good friend of mine, Neil Peterson, has grown 1,800 trees in his lifetime and sampled the fruits from every tree. He's picked out the 3 best trees for the best tasting fruit, and he just recently, last month, got a patent awarded for those trees. So he's got the 3 best, so he's got like the red delicious variety of pawpaw. Okay, so, and these will be commercially available someday. You wait and see. And he's got Snapple beverages and, you know, ocean spray, cranberry juice, and everybody wanting to make pawpaw drinks and things. Scientific name is Asimina triloba, and interestingly, a fluid extract of the seeds was sold by Eli Lilly Company as an emetic back in 1898. An emetic is something that makes you vomit. That's back when the physicians would make you vomit and they would bleed you and they would purge you to get you well. And as you vomit, the next time you vomit, remember that you sweat just before you vomit. That's called the diaphoria, the sweating. And as you sweat, you're sweating the toxins out of your body. And that's what they believed would help to make you get better, and probably does. The next slide. This is the distribution area of pawpaw all over the eastern United States. So this is not a rare tree. And I've got assays or population studies from the U.S. Department of Agriculture on how many pawpaw trees there are out there. And there's millions. Indiana has 18 million trees, for example, wild pawpaw trees growing. And they've got a few more because I've got about 2,000 of them planted on my farm in Indiana. So hopefully they're going to grow up and I'll make some money from selling the biomass there. And incidentally, we've created a new crop with this. And people at Kentucky State University have gotten grants about $170,000 a year to study the growth of pawpaw as a new crop to replace tobacco. Now, wouldn't it be neat to have a plant that replaces a carcinogenic plant? So, the next slide. Well, this is in the Annonaceae family then. There are about 120 genera in this family, about 2,100 species. Almost all of them are tropical or subtropical. So pawpaw is the only one that's temperate that grows where it loses its leaves. And so it's probably been carried northward by the Native Americans over the period of centuries, so that it's up here now in the deciduous area. But all the rest of them are small trees and shrubs that are in the tropics. Next slide. This is one of them that's related to pawpaw. It's called Annona muricata. It's also called graviola, or guanabana, or soursop, or Brazilian pawpaw. And they're not supposed to use the pawpaw name, but they're copying the pawpaw name in order to cash in on my good work on pawpaw, you see, with this plant. We looked at different graviola capsules which are out there for sale, and one of them was 24 times less potent than pawpaw. Another one was 56 times less potent than pawpaw extracts that are sold by Nature's Sunshine. So don't get misled by the graviola business. There's no biological standardization. I've done no research on this. Over 28 compounds I've isolated from this plant. There's a single ring compounds. They're not biologically active, at least 10,000 times less potent than the pawpaw compounds. But they're still active, but they're just not as potent. And so don't be misled by this plant. The next slide. So here's the Annonaceous acetogenins, and these are these active compounds. I know I don't want to bore a general audience with chemistry, but that's what I do for a living, you know. I bore people with chemistry. And so down at the bottom we see a structure. And if we start over at the right, you see a number 1 there, 2, 3, 2, 4, and then come clear over here to 34. So there's 34 carbon atoms in a row, all put together with 2 carbon atoms at a time from acetates by the plant. That's a long-chain fatty acid. And the only other place I've seen a long-chain fatty acid like that in nature is in beeswax. Other plants usually have only like 18 carbons in a row, like oleic acid in olive oil or stearic acid in lard. Those are only 18. Fish oils have some C20 or C22 compounds, so DHA and EPA they call them, and they're good for your brain. Fish is good brain food, and but these long-chain fatty acids are highly unusual. The only plant family that I know of that puts these things together. So there's C35 or C37 total carbons. There's a 3-carbon unit that adds on. They end up with those rings in the middle of the structure. Those are called tetrahydrofuran rings. Sometimes there's a 6-membered ring that's called the tetrahydropyran ring. And in this case, this compound has the rings non-adjacent to each other, but in the next slide, we see some rings that are adjacent to each other. Although this one's very weak, and the way it's showing up at the top there. But at any rate, there's a whole diversity of these structures in the plant. And people get the idea that a plant just has one active compound. Like opium has got morphine. It's also got codeine and papaverine, but morphine's the main thing. You make heroin out of morphine and all that. Coca plant has got cocaine, okay? Pawpaw has got 50 compounds, okay? So the next slide shows us some of the diversity of these compounds. These are adjacent ring compounds. Those letters A through E there refer to 6 chiral centers on that part of the molecule. Overall, there's 8 chiral centers. Those are carbons with 4 different things attached. And every time that happens, if you change 2 things around, you create a new molecule. So just with those 8 chiral centers, there's 256 possible isomers of the structure illustrated there. And it was our job to figure out what is the exact structural stereochemistry. How does this thing really look in 3 dimensions? And we did it. And it was published in the journal American Chemical Society, which is the most prestigious chemical journal in the world. And it's published right here. I was out there this morning, had a meeting at the American Chemical Society headquarters here in Columbus. Okay, so asimicin, trilobicin, biloticinone, these are compounds that we found in pawpaw. The next slide. This gives you a little hint as to the complexity. All these little peaks are tracings from high-performance liquid chromatography. And you get a peak for 1 or more compounds. And so one of my students, Dr. Zhao, isolated every one of those compounds and proved their structures. And in other fractions, there are more compounds. So in all, there's more than 50 pawpaw compounds. And we just were looking at an HPLC mass spec tracing about a month ago, and my goodness, I looked and I said, there should be some compounds that weigh 620 instead of 622. And we looked and there are compounds there. So there are more compounds there that we haven't isolated yet. So it's very complicated. Now, if you want to make a drug, drugs by almost definition today are single chemical entities. Only one compound they want in a drug because the assay is easy. Okay? And they can quantitate it, and they can determine blood levels and do all sorts of wonderful things with pharmacokinetics and things that I don't really understand and I don't even think are necessary, but they want to do it before you can get anything approved. And by the time they get done with all their tests, they've spent $900 million before anybody can even use the drug. And in the meantime, 600,000 people are dying per year, right? Where's the urgency? There isn't any. So fiddle on making a pure compound. I isolated pure compounds. I got the patents on the pure compounds, Purdue does, but we'll never get them developed. I've been there. I've been to Merck, Bristol, Lilly, Upjohn, Abbott, you name it, I've been there. They're interested. They take notes. They steal my ideas, but they won't license it because they didn't find it. It's a not invented here problem with those guys. The next slide. So here's this little brine shrimp test using these little sea monkeys. And we determined the LC50, that's a lethal concentration to kill 50% of the shrimp. And this has been the workhorse of my laboratories. And we still use them. We use them out in Nature Sunshine products to standardize the extracts. The standard extract kills brine shrimp at 0.5 parts per million with 0% moisture. That's the standard material. And there's 12.5 milligrams of that per capsule. And if it turns out a little bit less potent, I put a little more in. A little bit more potent, I put a little less in. So that from bottle to bottle, you're going to get the same effect from capsule to capsule. Next slide. Okay, then I could look at the different parts of the plant and see where the activity is the most concentrated. Initially, we're working on the bark, and the bark was quite active in the brine shrimp, but turns out that the twigs were the best, like 0 or 0.04 parts per million, with a yield up, about 1.78%. The leaves we see down here are 53, almost 54. So the leaves are like 1,000 times less active than the twigs. So I don't want any leaves in my extract, okay? Because they don't contribute much. And you see the unripe fruit there, 0.06? That's why I got sick, because I ate some green ones. And the plant doesn't want you to eat green fruit because the seeds aren't ready yet. So it makes the fruit, so it makes you throw up. So possums and raccoons and little boys won't eat them, until those seeds are ready. And then you can eat the seeds and go poop them out someplace and you spread the species around. Okay, so nature has this all figured out. Okay, the next slide. We also saw, I sent this extract off to Eli Lilly in 1982, and there were 2 guys there that believed in me, and they were both entomology people, not chemists. And so they tried the extract against insects. They had 7 insects in a panel, and we were able to kill 5 of the 7 insects. And this just shows you the results. FO2O is the pawpaw extract. You see that we're killing 50% of mosquito larvae down at 1 part per million. That is really potent. And also, blowfly larvae, killed 100% of them, and Caenorhabditis elegans, or C. elegans, is a worm, and we killed 100% of those worms. And so we knew that we had something that's biologically active, against pests, if nothing else. The next slide. So because of that activity, I decided, when I first went to Nature Sunshine product, 4 years ago, to make this into something that would be useful and get some cash flow coming. And the best insects I could think of to kill that would help people were head lice. Because head lice have become resistant to the pyrethrins that are in nix and rid, okay? And so these head lice then we could kill. And so we made this pawpaw lice remover shampoo. And I thought, boy, there's 16 million people. I did all the market research. 16 million people per year get head lice. Most of them kids, and most of them little girls, okay? So I said, we're going to help these people. It didn't sell. I published a clinical paper in it. I had 4 nurses do a study with a bunch of school kids. 100% effective. Got rid of the head lice, no problem. Can't sell it. You know why? Because I put lice on the label. Nobody wants to buy anything that says lice, right? There's a stigma against having lice. So I learned something. But at any rate, it's finally taken off a little bit. We're selling about 500 bottles a month right now, so it's going to take off. And people are out of desperation using this. Next slide. And also, we put this into a product that kills worms, because we killed Caenorhabditis elegans, and Merck tested 20-some of my compounds from pawpaw against Haemorrhagis contortus, which is a worm that dogs get. And it came in second to ivermectin, which is our heartworm product. So we knew that we had something there that we could use to treat worms. So we put a capsule pawpaw in this cleanse product, and boy, has this been selling great. 5,000 to 6,000 bottles a month we're selling on this one. I had no idea that so many people had worms. The next slide. So why do we use the twigs? I've already explained that to you, because they're the most potent plant part, and it allows the tree to survive. About 3 weeks ago, I gave a talk in Philadelphia to the Plant Conservation Alliance. These are a bunch of tree huggers, man. They were going to get me. Because, we're going to eradicate this pawpaw species and all this kind of stuff. And I prove to them, hey, by collecting the twigs, we preserve the tree. We're not collecting bark and killing trees and all this kind of stuff. So they were pretty happy when I got done. But notice that there are no leaves on these trees in the background. That's because I was really stupid and we were collecting the stuff in November. And I collected all that material, over a ton of material, and I made a huge extract and sent it off to the Canadian Forest Service for use against that spruce budworm, and it didn't work. Now, what in the world happened? So then I had to go back and do another experiment that took a whole year. That's why they call it research instead of search, okay? Because you have to do it over again to repeat it, okay? So the next slide shows us this little dotted line which goes up and comes down again. These are twigs from one tree collected each month over a year, okay? And the biological activity peaks in the 5th and 6th month. So that's in May and June. So you got to collect this stuff in May and June. And I collected other stuff in November. See where it is, 11 out there? See how low it was? No wonder it wasn't any good, see? So people don't understand this. Plants vary in their concentration of their active components. And you got to pick out the right time. So we've figured this out now. So we collected 110,000 pounds of pawpaw this May and June. 110,000 pounds this year. And we haven't put a dent in the wild populations. And as a matter of fact, the collectors are now out there, cutting down the hackberries and the box elders and the other stuff that are competing with pawpaw to grow more pawpaw trees in the wild. The next slide. Now there's another potential variation, and that is the tree-to-tree variation. So I went to Neil Peterson's plantations, and I collected actually 667 twig samples in one day. So I wouldn't have any variation on the day. And we only got through 135 of those because it took one day to assay each sample. But I got through 135 of those samples to see which tree was the most potent. And the difference in biological activity from the worst tree to the best tree was 1,000 times. So on the 4th row, the 11th tree was a real winner. 0.03 for the LC50 and the brine shrimp. But the 2nd row, the 32nd tree, was a loser. 32. The difference between those two biological activities is 1,000 times. You wouldn't think that to be that much variation from tree to tree of the same species of plant. But it's genetics. Some trees are really high producers and some trees aren't. So on my 2,000 trees that I got in Vermilion County, Indiana, my plan is to take some buds from number 411 there and graft them onto my rootstocks, and I'll grow tops of the trees that are highly biologically active. Okay? So this is what you can do with science. The next slide. How do these things work? Well, there are two sites of action, and they work by depleting energy in cells, whether it's an insect or a cancer cell or a worm. And they do that by inhibiting the mitochondria. Mitochondria are little football-shaped things inside of cells. They've got lots of membranes, and these membranes contain complexes 1 through 5 of the electron transport system. And the end result of these complexes working on breaking down your glucose metabolism is to convert it to water and carbon dioxide through Krebs cycle, and then make ATP. It's adenosine triphosphate. So ATP is a triphosphate. It's got 3 phosphates on it, and it's a highly energetic compound. So you can carry out reactions in your cells at body temperature, whereas normally in a chemical lab you'd have to heat them to make it work, okay? So they're cofactors. So ATP is the energy compound of cells. So we inhibit the mitochondria, and we know where we inhibit. It's inhibited in complex 1 of the 5 complexes. And Dr. John Casita at Berkeley has taken our material and tested it in subfractions of those mitochondrial complexes and found that it inhibits the PSST protein subunit of complex 1. That was published in the Proceedings of the National Academy of Sciences. That's the most prestigious journal in the United States, okay? So I know right where it works there. But also it works down in the plasma membrane, down at the bottom, by inhibiting an enzyme there called the NADH oxidase. And that prevents the cancer cells from growing anaerobically, without air, without oxygen. So we knock out the cancer cell's energy production in 2 ways. Anaerobically and aerobically. Okay? Next slide. Now, this ATP compound is also a precursor compound of other nucleotides, they're called. And these are UTP, CTP, and GTP. And also they're the deoxy sugars, which have deoxyribose instead of riboson. These compounds make up RNA, the deoxy ones make up DNA, which is in chromosomes, okay? And all of these are, in 3 days in leukemic cells, you can see are depleted. Like only 24% of the usual concentration of UTP is there if they're treated with Paw-Paw. Okay, these compounds are the building blocks of new cells for the new chromosomes and the new ribosomes, okay? So these compounds then, when they're inhibited, prevent new cells from forming. And furthermore, and this is almost serendipitous, we saw that an ointment made up of Paw-Paw prevents cold sores. And you can get rid of cold sores in 2 days with this stuff. Why? Because cold sores are a virus. And viruses are made up of these nucleotides. And by depleting the nucleotides, we have antiviral effects. And the world needs antiviral compounds right now. AIDS is a virus, SARS is a virus, okay? The common cold is a virus. So this Paw-Paw stuff is being used for shingles right now, which is another herpes virus, herpes zoster. Not only in an ointment form, but they're taking the capsules. And a doctor called me last Friday night and found me, you know, and I was still in Utah then, and she says, you gave me some of this jar of this little ointment, and she said, you told me to tell you the results, and I'm telling you it worked. I gave it to a guy with shingles. She said, orally and with the jar of ointment topically, she said, the shingles were gone in 9 days. And look how long people suffer with shingles, you know? So we've got something here, okay? The next slide. Now, here's another little angle. When you treat with chemotherapy, after a while it doesn't work anymore. You get breast cancer and it's spread to the lymph nodes. It's going to go to the bone marrow from the lymph nodes. You take the chemo, you go through all the stuff, you lose your hair, you get sick, throw up, and all this kind of stuff, and in 5 years you die. That's the usual pattern with chemo. Why? Chemo doesn't work anymore. Because the cells have become resistant to the chemo. And this little cartoon shows us why. There's a little pore that develops in the cell membrane of the cancer cell. It's a hole. And it's called the P170 glycoprotein. And this pore grabs onto the adriamycin that's inside the cell and goes and spits it out. And the next one. And it spits it out, okay? So it's a pump. It's an efflux pump, and it pumps the drugs out of the cancer cell so the cancer cell is no longer killed by the drug. And pretty soon all the cancer cells have got this pump, and you can't kill any of them anymore. And so it's called the multi-drug resistant pump because no drugs work. The Paw-Paw, fortunately, isn't pumped by the pump. I can explain to you chemically why that happens. But Paw-Paw goes in and inhibits ATP. And look at here in the middle of that structure, there are 2 sites that bind ATP. And ATP gives us the energy to drive the pump. So by inhibiting the ATP production, we thwart the pump mechanism. And just recently, it's been demonstrated in China, at least the Chinese are believing in what I've published. They've demonstrated that inside of the cancer cell, with the presence of the Paw-Paw compounds, drugs will now accumulate. Whereas before, with drug resistance, they wouldn't. And this means that we can restore the susceptibility of the cancer to the chemotherapeutic agent if you want to go through all that business. Okay, so I think it should be an important adjuvant to chemotherapy to help prevent the pump development and to treat cells that have already got the pump. Okay, so we treat resistant tumors with this business. Next slide. Now this just shows a slide from data from Lilly, from Abbott. And I wanted to show this slide because it shows the potency of the pawpaw compounds. The three compounds at the bottom, asimicin, biotinacin, trilobacin, are my best compounds from pawpaw. And they're way active in cell cultures here at Abbott, way out at 10 to minus 5, 10 to minus 6 micrograms per mil, whereas adriamycin's back here around 10 to minus 1, 10 to minus 2. So we're looking here at compounds that are like 100,000 to 1,000,000 times more potent than adriamycin, you know, on a molecule for molecule basis. So they're extremely potent. And I got data from the National Cancer Institute and their cell culture labs too. Works the same. Next slide. To prove the drug resistance business, at Wayne State University, they tried it against the M17R. That's a mammary tumor cell line. And the long bar going up reflects a very good potency of the pawpaw compound. And the little red bar at the bottom shows that adriamycin doesn't work to kill those cells, okay? But over on the right, we took normal cells. These are epithelial cells from the GI tract. And we see there that adriamycin is more toxic to normal cells than the pawpaw compound is. So why is this? It's because cancer cells are so voracious. Cancer cells are full, they take in glucose 17 times faster than normal cells do. Okay? And so they metabolize 17 times faster. So there's a therapeutic index of 17 there between a normal cell and a cancer cell. That's why we're not hurting the normal cells with pawpaw. We're in that index region. Okay? And then with the drug resistance here, those M17R cells have got that pump. And we're inhibiting the pump. So we're killing the cells. The next slide. Okay, then we went to some animal studies. And in this case, I got Upjohn's help. And I got Upjohn to help not because of the merit of all my terrific research. No, because Pat McGovern, who worked there, was a Purdue alum. And that's how stuff gets done today, isn't it? It'll make you sick, you know? Everything's based on politics and who you know. So in this case, I knew somebody, and that's how it helped. So Pat found out that in mouse leukemia, the L1210 mouse leukemia, these mice will usually die in 10 days. If they lived 12 days, then you divide T over C. 12 divided by 10 is a T over C of 120, it's called. So we had 139 with Taxol. So they almost lived 14 days with the leukemia with Taxol treatment. But the dose of Taxol was 15,000 micrograms per kilo. The dose of biotinacin up above was 50. So the difference there is 300 times. So the pawpaw compound is 300 times more potent than Taxol. But look at the weight change over there, grams per mouse. The Taxol mice lost 2.5 grams in 10 days. They only weigh 25 grams. So they lost 10% of their body weight on Taxol. Those mice are sick. If you've ever taken Taxol, and there probably isn't anybody, maybe a few people have, okay, makes you sick, okay? So Taxol is not a very good thing. But look at the middle compound, a 44% increase in lifespan for biotinacin, and the mice gained 1.1 gram. They gained 5%. So on pawpaw, they're gaining weight. Next slide. Now, this study was done again by Pat McGovern at Upjohn with athymic mice. These are mice that are so inbred they don't even have hair. They're called nude mice, and they don't have thymus glands. So they can't reject anything foreign. So you have to grow them in a germ-free atmosphere and feed them sterile food and sterile water. But you can take a human tumor and put it under the skin, and the tumor will grow. And in about 50 days, it'll kill the mouse. And so this gives you an opportunity to treat that mouse with something and treat a human tumor, right? So we took Gilda-Radner cancer, A2780 ovarian cancer, and put it under the skin of the mice and treat the mice for 10 days and then measure the tumor sizes and see if there's a tumor growth inhibition. Cisplatin at the bottom was the positive control. We had a 78% tumor growth inhibition with cisplatin, but they lost about 5% of their body weight. Cisplatin is a nasty drug. People have to be taken kicking and screaming back into the doctor's office to get the second dose because it makes you so sick, okay? But look at biotinacin, which is the first compound I showed you. 75% tumor growth inhibition, pretty close to 78. And the mice didn't lose any more weight than the vehicle control caused. So, hey, this stuff's working. So this study was $66,000, the information on that slide. Next slide. All right, these things work by causing programmed cell death. And what we see here is a DNA ladder of fragments of DNA. On the right, we see the standard of the DNA fragments. On the second from the right is cells, the DNA from cells that are killed with heat, which is called necrotic cell death, and same with the compound, or the column on the left. But the second in from the right, the column you see those little bars going up? They're very faint. But those are the DNA ladders that are formed from the DNAases cleaving the DNA in the cells as they're being killed by Paw-Paw. And that's called programmed cell death. And that's a normal type of way to kill cells. We don't want to just kill the cells like burning them away and leaving a hole there. We want them to be resorbed back into the system. A good example of programmed cell death is a tadpole's tail. Tadpole develops into a frog. The tadpole has a tail, the frog doesn't have a tail. Where did the tail go? Did it fall off? No. It got resorbed back into the frog system. That's what we want cancer to do, get resorbed, okay? And that's what Paw-Paw causes, is programmed cell death. Next slide. All right, just to show you that we've had all kinds of scientific background here. Here's the cover of the journal Natural Products, which is published here at the American Chemical Society, along with the American Society of Pharmacogosy. And we see the picture of Paw-Paw with the structure of my compounds at the bottom and a butterfly. And this was on the cover of that journal for 6 months back in 1999, and it honored my research, okay? And I'm very grateful for that. But I want to also mention the butterfly. See that butterfly on there? This is a white and black butterfly called the zebra swallowtail butterfly. You know, basically I'm a scientist. I like to answer questions, crazy stuff like, hey, this butterfly has been eating my Paw-Paw plants. It eats the leaves. It's an obligate parasite on Paw-Paw leaves, this butterfly is. And it's supposed to be an endangered species, and it is endangered when it gets on my plants. And it has this weird looking little caterpillar, you see, that eats up your leaves. And it didn't read my papers that said Paw-Paw are pesticidal, you know. So this is a good example of evolution of the insect staying ahead of the plant. So this plant made these compounds which are supposed to keep insects from eating the plant and to keep beaver from eating the twigs and cows or deer from eating the leaves. And this insect has figured out a way to grab onto those compounds and use them in some way. So I want to know, what is the chemistry of the insect? So I got the world's expert on zebra swallowtail butterflies. He collected me a bunch of butterflies. We freeze-dried them, extracted them, and assayed them. They kill brine shrimp. Aha. So then we assayed them with our chromatograms and got those peaks. There are the Paw-Paw compounds in the butterfly. So the butterfly eats the Paw-Paw and concentrates the compounds. Now, if you're a bird and you eat that butterfly, what's going to happen to you? You're going to throw up. And so birds throw up if they eat this butterfly, and so these compounds give the butterfly a natural protection. And that's an example of chemical ecology. The next slide. So these four mechanisms and summarize how Paw-Paw works, and I don't want to go back over these again. I'm just taking up your time. The next slide. I want to point out then we went to beagle studies, and I figured that this stuff wouldn't be toxic because beagles should throw up. And sure enough, that's what happened. We got up to 32 capsules at a time, 4 times a day. That's 120 some capsules per day, and the dogs just threw up. They had a little watery diarrhea, they threw up, and they went their merry way. They gained weight just like the control dogs did over several weeks. And at the end of the experiment, Dr. DeVere said, Jerry, there's no need to sacrifice these dogs. I know they're okay. There's nothing wrong with their livers, I can tell. So he said, I've never seen an anti-cancer substance that wasn't toxic, but you've got one. And this was an FDA-approved lab. So pawpaw is very effective and exerts very little toxicity. The next slide. Okay, so then we went to people, and this was a big step. You give something to a person, you don't know what's going to happen. So I looked at the dog data and I calculated, well, okay, the dogs can tolerate this much, people ought to be able to tolerate that much at least, and people are tougher than dogs. Guess what? They're not. So the first doses that we gave to 3 ovarian cancer patients made them all throw up. So I cut the dose in half, and they could tolerate it. And the first lady in 1 week cut her CA-125 levels from 3,000 to 1,000 on pawpaw. That's the cancer antigen levels. And it started to look pretty good. Then I got Dr. Forsyth over in Reno. He's the director of the Reno Cancer Screening and Treatment Center. And he said, okay, Jerry, I'll give you 20 patients. And they'll be terminal. Because in Nevada, if you're a terminal cancer patient and you and your doctor agree, you can take anything experimental that you want to. And you don't have to go through an investigational new drug application or a board at the company or any of the stupid stuff that the federal government set up to try and protect people, okay? People who are going to die anyhow, okay? And so we tried the 20 patients. That was 2 years ago. Out of the 20 terminal cancer patients, all of whom we expected to be dead in 1 year, 7 have died and 13 are still alive. And those 13 people are stable and they're living good lives. They're living with their cancer. And I used to tell my students at Purdue, I lectured on I lectured on cancer for 30 some years, 28 years at Purdue, and I'd tell my students, what's the definition of life? And I get all these esoteric definitions. No, life is the interval between birth and death. That's it. Life is an interval between birth and death. And what do? With all the good things we try to do with healthcare, is we try to extend the interval. Nobody in this room has an indefinite interval. Nobody's going to live forever. If we can extend an interval for somebody, we're going to do some good. So I got some folks now that have been kept alive several months beyond when they were supposed to be alive. Just a couple of weeks ago, we lost a guy in Poland with brain cancer, but he was supposed to die in February. We gave him several months. And in this wellness report that was passed out, this little newspaper-like thing, there are 19 case studies in there of people from our study of 94 people over the past 2 years, with them giving their testimonials on taking pawpaws. And a couple of them actually have viruses. Even MS, multiple sclerosis. A lady can now write with her right hand, and she couldn't before, for 3 years. Took pawpaw 1 month. And that helps to prove that viruses probably cause MS. Hepatitis C, we've had some good results with that. And we haven't got studies to prove all that, but it's going to happen. And people are taking it even for the common cold. And it seems to be helping because they're antiviral. Okay, so just a few of these results here. Some breast cancer patients. So let's take patient number 8 there. That was Nona, one of our secretaries. And I saw her crying at the picnic. She says, I got breast cancer. She's crying, you know. And so I said, well, hey, in October, this was in 2001. I said, in October, we're going to have this pawpaw stuff ready. Do you want to try it? Be a guinea pig? She said, yeah, I'll do that. So she tried it. She took it, and then she took chemo 1 round, and they couldn't find a tumor anymore with MRI. So they gave her 5 more rounds of chemo, and she lost her hair. She got sick, you know, and all this stuff. And it came up until June. That was in December. She got up until June, and then she went in to the surgeon. The surgeon had to get his money. So he looked her over, and he couldn't find a tumor either. So he did an ultrasound. He said, oh, I think there are 3 little blips there. I'm going to go see what they're like. So he went in there, and he cut them out, and he took the whole left breast. And I said, I want to see the path report on this. And sure enough, the path report said, okay, traces of ductal carcinoma. So, okay, maybe he was justified in taking the left breast. Well, then they followed it up with radiation. And all the time she kept taking pawpaw. Okay? And so by September, the time September rolled around, she'd been through all this stuff. and they couldn't find any traces of tumor in her. 14 subaxillary lymph nodes did not have the tumor, and so she has a sign on her desk that says, "I'm cancer-free." So I think that was pretty good. Patient number 12 with breast cancer is 72 years old, stage 4. One of Dr. Forsyth's patients, been through chemo, nothing was working anymore, so she's refractory. It's getting worse. And so she put on Paw-Paw for 6 weeks. That's the only thing that was any different, just taking the Paw-Paw for 6 weeks. Her tumor marker, the CA2729 marker, went from 160 down to 80 in 6 weeks. So we cut the tumor antigen level in half, and the size of the tumor is reduced significantly. The next slide. Okay, with prostate cancer, number 48 was an old guy, 85 years old, and he wasn't a good candidate for surgery. So they put him on Paw-Paw, and in 6 months, we kept his PSA level, essentially level, from 36 down to 30, which is pretty good. But then at that point, he got allergic to it, and he got itchy, so he quit taking it. And I actually met him. And let's see, patient 56 had stage 4 cancer. He's in North Dakota, and he had a huge tumor in his left neck from prostate metastasis. And one growing on his hip bone and one in his abdominal cavity that they detected with x-ray. And the radiologist called Dr. Forsyth and said, "What's going on with this guy? His tumors have reduced him in mass by 25%. And he'd been taking Paw-Paw for 6 weeks. And he was only taking 2 capsules instead of 4. He didn't get the instructions right, you know? And so that looked pretty good. And the last one there on the PSA, this prostate-specific antigen, was my eye doctor. This guy got prostate cancer, and it was an aggressive type. And so he started taking Paw-Paw, and he did the radioactive pellets as well. But anyhow, he went from 4.5 to 1.7 with the radiology and the Paw-Paw. So he's pretty happy. He still charged me for these glasses. So. The next slide. Okay, this patient was from Australia, and she had lymphoma for 6.5 years and gone through 3 rounds of chemo, but the chemo didn't work anymore. This resistance business had come on, so they developed that pump, you know. And her white cells were going up, and on 35 days on Paw-Paw, we knocked the white cells from 34 back to 11. And the lymphocytes were the ones that were going crazy, and we knocked them from 67% back to 30%. And both of those values are back in normal ranges. So against the soft tumors, like lymphomas, we have good results. I don't have good results with leukemias because I haven't had many people with leukemias try it. But it worked in the mice with leukemias and extending theirs. I think it's going to work with leukemias. And we got some little kids, incidentally, like sat down and calculated doses out for them with leukemias. The next slide. Okay, lung cancer. Yeah, this guy was one of Forsyth's patients as well, and his antigen level went from 275 down to 222 just in 2 months. And he gained weight, and he'd been in bed in a wheelchair before, and he's walking around now. So we're extending his interval, right? And he'd previously been on 2 years of chemo, and chemo wasn't working anymore. The next slide. This guy's picture is in that wellness report. He's got a picture with a horse in there. And his name's Eddie. And in December of last year, he was supposed to go home and die. And he'd had melanoma, that's black mole cancer, from his arm. He's a farmer in Oklahoma, and it had gone to his lungs. And he had these masses of this stuff growing in his lungs. So the doctors sent him home, and he couldn't breathe, burned to breathe. And in 4 days on Paw-Paw, he could breathe again without burning. And then his strength returned, and he's out. He just wanted to go see the cows one more time. He got up and testified in one of these meetings. He was there, you know. And it brought tears to everybody's eyes. You know, Dr. Keller said about half the audience was crying when he got done. And so Eddie can now ride a bicycle and walk, and he goes down and sees the cows every day, and he got out and plowed with the horse this spring. And also 2 lipomas, which are little fatty growths, little fatty tumors on his arm, they disappeared. And another interesting thing is that his toenail fungus that he's had for 10 years has gone away. taking pawpaw. So it's antifungal as well. The next slide. So this just gives us a summary. Alkaline phosphatase, that's an indicator of bone cancer. And I got a big hug from a lady in East Lansing, Michigan, who was on our study, and she's back in the normal levels with the alkaline phosphatase with bone cancer and raising her kids and carrying out a normal life, and she's supposed to died last year. So now, no hair loss, no bone marrow depression, no GI bleeding. And we've seen benefits with cold sores and shingles and toenail fungus and MS and acne, athlete's foot. I got it in a lotion for paint, you know, for kids' faces. One of my workers in the lab was in there. I caught her in the lab making up more pawpaw lotion. What are you doing this for? Oh, I'm giving it to the kids. She had all the teenagers in her neighborhood are using pawpaw lotion for acne. It's working great, you know. Psoriasis, eczema, all stuff. Unfortunately, we're having trouble getting it into the lotion and salve form because they want to heat it in the manufacturing process and they kill the activity. So I've threatened to go there and make it myself by hand so we can sell it. The next slide. Okay, so this is the pawpaw story, and that's where we are with it. And there's handouts of the slides in the back in the table if you're interested and want to see that. And I'll answer questions if I can. Thank you very much for your attention here tonight.